So I'm actually glad I got to follow the previous talk, because we have seen a lot of similarities between what was just described and how we approach antibody validation. So I'd like to thank the organizers first of inviting me back, actually, to give this talk today. So we're going to really go over all the different methods that apply to antibody validation, and that's where the title of my talk came from, because it is really a method of applying scientific approaches that have been used for many, many decades, obviously. New approaches that are always crucial to add to your validation, as well as just common sense. And I'll show you a couple examples of where certain things just come back to us from customers and how we approach some of that feedback. So first, let me start off with a schematic of these are all the pieces that really make a good antibody validation strategy. I'm going to say, just in general, these are all the pieces that really play into that. One of the most important parts we still believe in is the immunogen design. If the immunogen design is not done correctly right out of the gate, you will not get the right antibody out at the end of it. So we've spent obviously a long, long time on full-length proteins. That's the life's blood, so to speak, of our company. And we spent decades on just making full-length bioactive recombinant proteins. And the feedback we've gotten more and more and more over the last couple of years especially is that those are truly making some of the most specific antibodies for certain applications. Again, I completely agree with the statements made before. It's a fit-for-purpose reagent. It's not a one-size-fits-all. And I think sometimes that's being a little bit overlooked, I'm going to say, in some of the public discussions. So when you have an assay target, for example, it is very, very crucial you start with a full-length protein that is fully folded, unless you want to exclude specific isoforms, things like that. The way we purify our proteins, we can pretty much tell throughout the process when we start losing the folding. Obviously, we add bioactivity measurements at the end to really make sure that the protein we start with has the best potential folding to give you the antibodies you're really requiring for any immunoassay related approach in the end. Other designs require obviously more surgical approaches, so peptides is definitely our second go-to tool, but then there's also still whole cell immunizations, things like that for very different difficult targets. And after you make your antibody, you do need biologically relevant models, obviously. We use all kinds of tools to figure out what is the best model to use for a specific target. A lot of scientific literature has come out over the last 10, 20, 30 years of science, obviously. And when you've been in the business as long as we have, you really need to go back, look at the literature again. Is there more known about the target? Because that can drive some of the retesting we're doing year in, year out. So our catalog is not static. We keep going back. There's more insights on specific targets. And we actually retest then antibodies, and we sometimes find, based on the literature these days, the specificity is probably not given in any application. And then we make decisions on whether or not we're going to continue carrying that product, or if we're going to discontinue that. Just to give you an example how we use that. Knockout cell lines, you've already heard some about that. It is definitely a very good tool to use. It's quick for Western blots. It's also very, very useful for ICC and flow, but it clearly doesn't apply to IHC. Optimized and transparent protocols. Us as a manufacturer, we find very, very important that we share the protocols to the detail that customers can actually reproduce the results, so that the scientist using our antibodies in the end gets the same results that we've been able to get in the lab. So the transparency of the protocols for each application is very, very crucial part of our process. Obviously, you need to understand the target. Again, this goes back to you need to keep looking at the literature. You need to see is there more insights into specific models that we should be using. Are there specific treatments? So biological model validation is one of the most crucial parts still of antibody validation, and it will stay one of our mainstay approaches to that. Full product traceability. So when we make one, it's nice, and when you make it once, that is good. But you need to make it over and over again. So this goes back to one of the tables, for example, on polyclonals, monoclonals. How do you make sure that what you make once is reproducible for the next generation to come, essentially? So We're planning always ahead. If we can only make it once, we have no good method to keep making the same product, keep validating it the same way. We're not releasing it. It's as simple as that, because I cannot guarantee that we can deliver the same quality over and over again. We also need stringent QC parameters that go alongside it. That is pretty much going hand in hand with the full product traceability and just delivering that reproducibility. And then you have a clearly defined product at the end. You disclose it in your websites and inserts, things like that. So we're trying really to put out as much data, as much information as we can around all that. Once it's released, we do continuously incorporate customer feedback. We're only so many people. We're not covering the globe, but luckily we have access to a lot of really, really good researchers. They give us feedback on how they've used antibodies, what they found with them, what they like, what they didn't like. And then we incorporate that, and we have a lot of back and forth with customers and just really making sure that we're meeting the expected outcome or expected data. So this is really one of our core principles, is we're guided by ISO 9001. The whole company is under the ISO 9001 umbrella. Parts of it we also have to adhere to ISO 13485. That's the higher standard that goes more towards the GMP type of product line. And then everything that is done on the protein side, on the antibody side, as well as the assay side, as one of our internal customers, has to adhere to the same standards. That's just the underlying foundation for how we deal with all our products. So we have about 13,000, 14,000 antibodies, but all of them have been made in-house. What that gives us is we have all the data that go alongside that over the many, many years that we've made antibodies. So that's what allows us really to go back and check, is there more information available? Did we make a mistake during development 20 years ago? Did we overlook something? So that is really, really crucial for us internally. Again, we are using a lot of our bioactive proteins, and we make sure that they are truly bioactive and not just they meet the size criteria that we're after. We do more and more whole cell immunizations. That becomes important on more the receptors, full-length receptors especially. Obviously, you cannot always make just the extracellular domain. So those get very tricky, because then you need to design your whole testing at the back end really around excluding the non-specific reactivity that you will get. That's a given. So that is much more involved testing than at the back end, and that's why I say when we start an antibody, we really design the whole system around it and not just one part or the other part. It all really needs to go together. Peptides, again, mostly used for phosphorylated targets, but also more and more used for very specific isoforms you want to detect. Some of the interleukins have specific subtypes, A's, F's, and we only want to detect one or the other. There are disease models that really require one or the other, so that's really when we default to peptides to really make sure we have the specificity at the end that customers expect, and we can say this is the epitope that the antibody recognizes. It's very crucial in those particular cases. And then we're also doing more with haptens, but I'm not going to go into details today. That's small molecule work. It's a very different beast, I'm going to say. Because then you're starting to look at really, I have a structure with a double bond here, I have a structure with an extra benzene ring here. Do I need to check all those and cross-reactivity? So that is really where the specs up front become very, very crucial, and we're partnering with our Tokris colleagues on those ones, because they have the chemistry expertise, and that's really what's needed for some of those types of targets. I show you one example today on a tag, and it's partially food for thought, so we'll go into that later. Once we've made all that, we made all the decisions, we set all the specs, we do our work on the antibody side, then obviously we need to make sure that all the applications that are applicable to the target are being tested, and then we come back and really cross-check that they do overlap. So this is really making sure that you pick the clones that have the highest validation data behind them. We have a lot of, obviously, internal customers, but we're mainly working for all the applications that have been used by all the science communities, going from Flow, Western, ICC, IHC. We also do a lot of blocking neutralization testing, so we have multiple assay types. around that. And again, this is where your biological models become really, really crucial, because for blocking and neutralizing assays especially, you need to make sure you use the right model. So these are our models, that our antibody types that we are developing. That's our mainstay. We have obviously hybridomas, and then we also have recombinant monoclonals for both rodent as well as rabbit, as well as our polyclonals. That's goat and sheep. The validation strategies, the validation level is the same for every single antibody. The reason why I bring that up is really the quality of an antibody isn't driven by the type, it is driven by the validation. You can make very, very good polyclonals, or if you don't get your validation strategies at the back end correct, the polyclonal will give you what you put in, which could be very poor quality in the end. So every single specification and validation you have to apply to a monoclonal has to be applied to a polyclonal to the same level. Once you make them, then obviously the reproducibility and your QC and manufacturing parameters become even more crucial on the polyclonal side. So there is a different level when we're talking about polyclonals on the manufacturing end, because you really need to ensure you get the same lot-to-lot consistency. So you need to make sure you test all the applications again, and not just say, okay, it's the same clone, because it doesn't exist in that case. All the applications, again, get cross-checked. So a group of scientists sits together and they all compare the data. And it can take hours sometimes to hash out certain clones, whether or not we really truly think the data is specific enough to release as a catalog item. So that part we take very, very dear to our hearts. And that's when some of the decisions have to be made, whether or not we need to go back and actually check more models, whether or not we even want to go ahead and release it at all. And then we also at the back end, we can sequence verify a lot of our antibodies now, so that is pretty much applying to every single monoclonal. We've sometimes gotten questions on, do you really have a monoclonal? Do you have maybe two species in there? Do you have a biclonal? For most of those questions, we have a very easy way of just sequence verifying that and say, yes, we have a true monoclonal, or this is an old clone. For whatever reason, we have a biclonal, or sometimes you do have shifts. So we have very easy ways of addressing that very, very quickly. So 5-pillar validation is a little different from what Matthias was just showing. So maybe we do have a sixth one. We can discuss that later. So we obviously take the genetic strategy. The mainstay is their knockout, whether or not you do CRISPR-Cas9 or you do an siRNA knockdown. We're also now partnering with our company, sister company from ACD. So they have an RNA scope technology, and I'll show you how that works. So that is our orthogonal approach for the IHC, and I'll show you example data today. We just started that recently, and we're going to leverage that more and more. Independent, clearly one of the things we are doing all the time is we have many antibodies to many of these targets, so we do the cross-testing. And then we're doing expression of tag proteins, not quite so much. We use it more on very, very difficult targets where we really have very few options left over. And then definitely biological strategy. So that is what we consider our 5th pillar. It's a standard go-to model, and it has absolute valid approach in antibody validation. It will be one of our mainstay, because sometimes you just don't have a choice. And in all honesty, it adds a lot of value on more the biological behavior of the target, rather than just it's there, it's not there. So this is an example on A431s plus EGF, and then this gives you the phosphorylation. And after the treatment with alkaline phosphatase, you remove the phosphor residues and you lose your staining, and this is particularly important to ICC. So I'll show you 2 examples of how we're using more novel approaches in our antibody validation. So we've obviously added knockouts. It is, as shown before, it's very nice to see, and very satisfactory actually, to see a Western where you have a clear band in one lane, you have the band completely disappear in the next. It is very, very easy to read. There is no questions left over. We've chosen a system that allows us to go into multiple cell lines. because we said we want to align which cell line we're knocking the target out in with as close as possible to what the research community is actually using. So this would be your HeLAs, your MCF7s, your HapG2s, all those cell lines. Because then we can also go back and say this is what we originally validated the antibody for, in which cell line model. We knock it out and we show the same cell line again, just now in a knockout model. So that's the approach we've taken, and we've now validated over 100 targets that way with over 1,000 products that applies to. We're going to continue this in FY19. This is just 2 examples, again, going back to polyclonals don't have to be a bad reagent. It really depends on how well you validated that the first time around. So I show you an example here. This is our anti-CD105, really nice, strong band in a HeLa cell line. After knockout, it's 100% gone. There is no questions. We're also staying with the same Western blot approach that most of our customer base is using, which is still an ECL-enhanced Western, rather than a direct fluorescence approach. That's just what we've chosen to do because we wanted to align as closely as what the bulk of the research community is still using in Western blot. And that way also, if you have any other additional bands that you see through the enhancement using an HRP ECL, you will still see that. It will be very, very close to that. And then loading controls is obviously GAP-DH. On the right is a model where we used a PC3 cell line and we knocked out MMP1. And again, you see the band in the wild type. You don't see the band in the knockout. Not all models will work in all cell lines. We had to learn that, obviously, but you also will see some large catalogs of knockouts. So there is just, there's some definition, some thinking that has to go into before you design your knockout. I've seen knockouts done in HeLAs for CD28. It's just not a good model. When you look at the mRNA profile that I'm showing here on the lower right, you actually don't even see on standard mRNA level that CD28 is present in that cell line. So I wouldn't start with a HeLa cell line as a knockout. So this can drive some of the decisions of which cell line we're using. Essential genes, we are using a system that allows us to knock out larger chunks of the gene. So some of the more essential genes that you would expect could be very detrimental to a cell, we've actually been able to address. I'm not saying it's very straightforward. The cells do grow very, very slow. It takes a long time, but we are sometimes able to address more crucial genes we find with that system. New fusion proteins can also show up. I show you an example today. We're in the process of sequencing exactly what that band is on the gel. But you do see those, and it is not just present in one antibody. We've got a couple examples where you knock it out, and we've compared 5 different antibodies from 3 different vendors across, and it's just a new species that shows up. So sometimes the cell just tries to escape the knockout by making a new fusion protein. It happens. High knockout percentages obviously can be achieved. Usually if you have a really good knockout to start with, you do see that 100% reduction. Knockouts can be designed to target specific isoforms. It all depends, again, on what your antibody initially was targeted for. And sometimes you actually start uncovering, oh, I have an AKT1 antibody, but when I knock out AKT1, the band is still there, and you find that it actually still detects AKT2, for example. So that's one of the nice things actually about using knockouts. You can start dissecting some of those antibodies much better. And then you just have to disclose, okay, it's actually a pan-AKT, it's not an AKT1 antibody. So you have to just tell your customer base that very clearly. So this is one of the examples. The initial data we had on this antibody is shown on the left. So we used initially, when we developed this, a biological model for validation. We used multiple cell lines that have been described to express STAT5B in high amounts. And we were able to reproduce that compared to the RNA data. On the lower left, we also induced a cell for ICC with interferon gamma, and you see the really nice signal resulting from the STAT5B. We then made a whole panel of the STAT5s and knocked every single one of them out individually in a cell line, because one of the things that came up during our validation and our testing is is one of the stats maybe compensating for one of the other ones. Would that potentially lead to cross-reactivities, things like that? So we said we're going to just design every single one of them with a knockout cell line, and we'll run them all on the same panel. And when you do that, for this particular antibody, we only see stat 5b really disappear. All the other stats are still present. On the right, then, we did the ICC, same knockout cell line. And again, it validates that the biological approach has an absolute space in antibody validation. It does not replace it. And it really goes back to you need to use more than one approach. It's not a one-size-fits-all on validation. You just need to combine methods, really review the data as a package, and then make a decision. This is the one that I personally like. SEMUC has been an interesting target. There's been a lot of literature about SEMUC on IHC, in ICC, as well as standard MIG-TAG. So when you look at the best known clone, 9E10, is a MIG-TAG antibody. We did test this in a SEMUC knockout. And what you start seeing is that you see the band disappear, but you see others appear. So that was kind of surprising. When you look at polyclonal, again, this is a MIG-TAG specific polyclonal. Again, you see the main band that you expected to disappear, but a new one appears. Again, we don't know yet what it is. We're working on that one right now. But the size between the poly and the monoclonal is the same. So the band that appears is the same detected by both antibodies. Now, when you look at an antibody that was made to the SEMUC protein, not the tag, you do see the knockout and it completely disappears. So when you have an antibody that you buy for a MIG-TAG as a design and you want to use it to detect a SEMUC native protein, I would say be cautious because it's really not what it was intended to do. So I wouldn't say that the SEMUC-TAG antibody is a bad antibody. It is just not used for the right purpose if you start using that clone for IHC, for example. And obviously a lot of people have used it for IHC. Personally, I have too. I spent 3 months using that until I finally said I'm not going to use it anymore. It was a learning experience, but it's all good. So it is really a fit-for-purpose reagent. If you have an antibody, look at the immunogen that was used, look at the description. Was it made for an anti-tag purpose, or was it made for a detection of the native protein? Very important distinction, especially for SEMUC. And then on the right, I'm just showing you one example where we ran a MIG-TAG protein just as another control. So if you have a MIG-TAG protein, in this case Wnt3a, you detect it very, very cleanly. On the left, there is no detection if you take the MIG-TAG out. And then we're starting to add RNA-scope now to our IHC validation. So this is one of the orthogonal methods that we're right now starting to employ more and more. So let me go over what RNA-scope is very briefly in case you've never worked with it. It requires target retrieval. The RNA probes are not small. You have to get them into the cells. You have to have a method right up front that allows the probe to go into the cells. Then you hybridize what we call a double Z probe. The double Z probe design gives you much higher specificity than a single Z probe. So that's really what the backbone is of the technology. So you hybridize that first level of double Zs. And then you have a couple more levels of hybridization steps to really give you the amplification. So this allows us to really get down to low levels of mRNA in the cell. And then you have your amplifiers, and you can either detect that in a fluorescent mode or in a bright field. I'm just going to show bright field data today because it's a little easier to present with that. And then you visualize, obviously dependent on which visualization modality you've chosen. You can also quantify it. For this purpose, we're not quantifying. That is more for the pathology side. So for this purpose, we're doing more qualitative comparison. So you can do this on 2 separate slides. This is an example for anti-CD68. Before we do this, we do a lot of IHC pre-work, so all the standard approaches you would take for an IHC validation of different tissue types, different retrieval methods, going through the optimization for the antibodies have been done at this point, and it met our criteria for it is a specific CD68 based on those results. And then we stained a serial slide with the anti-CD68 probe shown on the right in pink. And you get pretty much an overlapping pattern on the RNAscope and the IHC signal. Another example is alpha smooth muscle actin. Again, all the pre-work on the standard IHC validation has been done before. I'm not going to have time to show the data today. On the left is the IHC. On the right is the ISH probe. What you will see is that most of the expression levels between the mRNA and protein do overlap, but it will never be 100% correlation. mRNA and protein have very different turnaround times in the cell, so they break down certain rates, certain time frames. So we're not looking for 100% correlation on the levels. We're looking for the same location, same patterns. You can take it to an extreme level and really do this on the same slide. It is a very, very harsh method on the tissue because you need a harsher retrieval for the RNAscope, but it does give you the 100% confidence you're looking at the same cell. So on the left, again, we went back and used our CD68 in the human tonsil in this case, and you see the same two cells that give you an IHC signal represented here in blue also have a single dot in RNAscope. On the right is another example using GATA3. Same approach, same slide, using IHC on the same slide. And then we have been known for many, many years as making antibodies for receptors. So this is more where I'm going to go into a small case study of just giving you an example of how we work with our customers. We've made an antibody a while back to a receptor, and we were able to actually work with AstraZeneca and get a publication around this. So we're actually, AstraZeneca was actually able to then crystallizes and get this crystal structure out. So we're very happy that they used our antibody for this study. The thing with receptor antibodies, a lot of them are used in flow cytometry. So just in general, I'm not going to go through the whole workflow, but this is a rough workflow that you're looking at for flow cytometry. Every single time you have this type of workflow, you have a lot of potential failure points. Every single step of this can lead to false positives, false negatives. I'm just going to show you one, which is incubation time and temperature. CCR7 transmembrane protein changes conformation a lot in the membrane. They flip in and out of the membrane. They cycle in and out of the cells. So very, very fluid model. It is not a rock in a wall. And what you see is when you stain the cells, same cell prep at 4 degrees, you really don't see much staining with CCR7. This was coming from customer feedback. They were really struggling with CCR7. This is why we started looking at this. You bring the cells to room temperature, you start seeing a signal come up. Ideal staining is at 37 degrees. Again, it has to be optimized for every receptor. CCR7 works best in 37. Other receptors we've seen work better at 4 degrees. So it's really one of those things where you need to understand the biology, and that's why I say we're using common sense as well as scientific thinking. We're also partnering with the HLDA. This is our external validation. We're participating every time they make a round of new CD markers. And so far, we've been able to contribute 45 antibodies to the HLDA organization and establish the CD markers with our antibodies. We're following their process very, very closely, so as much as we can. We don't have access to all the clinical samples, but everything else that we can, we pretty much use the same process that the HLDA is putting out. And this was just one of the later papers where they established new CD nomenclature in 2015. Okay. Once we've got everything established, so circling back, important that we can reproduce it. The first lot gets the same, the second lot gets the same testing as the first, same with the third, same with the fourth. Always the same testing is applied to make sure we can reproducibly make these antibodies. So in short, our validation methods just simply have to be aligned with what we're looking at. We have to use continuously new methods that, as they become available, as we can make them applicable to our antibodies. And RNAscope is one of those that we just added recently. Again, that's going to continue, so we'll have more data around that and what that looks like. And then we are definitely dedicated to a 5-pillar approach, maybe 6. So it's what we've just, as a company, decided we're going to stand behind, and that's how we're going to continuously support all our products. Okay, thanks. Any questions? Yes.
Do you intend to apply only forward-facing for new antibodies developed, or you can do your entire back catalogue?
Yeah, that's, I'm going to say, where it becomes a little bit of business discussion. I would love to, but I have a feeling that our CEO wouldn't necessarily like the bill that comes with that. So yes, we are continuously going back and not just applying this to new antibodies. We've gone back over the last 3 years, actually doing a lot more first data edition. So that's the first thing, is the biological models are much quicker sometimes to apply, so we can do that on a broader scale. But like knockouts, we applied pretty much to every antibody that applies to that target, so we're not selective that way either. So technically, eventually, we'll get through the whole catalog, but it'll take a little bit of time. So.
Thank you for pointing out your successes with the correctly folded proteins as immunogens. That's also my sort of fairly limited experience. I just wanted to note that, of course, the relationship between mRNA expression and protein expression is not strong.
That can be correct.
It can be correct.
Yes.
It can be correct.
Yes.
But
yeah,
it's a lot of money to spend on an RNA scope probe, which I think costs about €15,000 these days, to, if you're not certain that there is a correlation. It's sort of like a circular argument. There's a lot of debate in the literature, as you know, about the correlation.
yes. Yeah, so again, in-house we can do it. I completely hear you. On the customer side, it looks a little different on the financial part. I'm fully aware of that. And SEMUC is, again, one of those where I've run into that, where the correlation isn't really tight. So again, it will not work for everything. None of these methods are a one-size-fits-all. But we will use it as much as we can to really give that extra level boost in confidence that the IHC antibody is specific. Because I can't address some of these things with another method as quickly, I'm going to say, as I can with the RNA scope probe. So it's really, you just need to see where it fits. But a negative result, yes, a negative result is you don't know. Yes, it doesn't make the antibody fail.
You can go first, but there was also a gentleman at the back.
Just on the IHC front. So, do you use positive control when you do the ISH to look for RNA degradation? And if yes, do you see sort of any correlation between loss of protein and loss of that positive control in your tissue?
So the tissue bank we're using has been pre-qualified for ISH. So we use a positive control probe first on the tissues to make sure that the tissue, because we have no control over the pre-work on the tissue itself, that has good enough quality and that the mRNA hasn't completely degraded. We have certain blocks where the control probe comes back completely blank, so we know something happened during the whole processing, maybe it's just laying around in the corner too long. So we would never use that tissue for this.
Okay, so you don't know if you would also see loss of your antibodies hitting that same tissue or not?
Correct. We don't even go there at that point because it didn't qualify for the ISH part. That's a good question.
That's the last one then. Otherwise I'll get shot by Andrew. Go on.
Have you come across a situation where you have an antibody that is validated by knockout strategy or by biological context?
I'm sorry, you have to repeat that.
Try to speak louder.
It's a lot of noise in that corner.
I was wondering whether you came across a situation where you had an antibody not validated by knockout but by biological context. And what would be your approach? Would you sell that antibody or would you consider it validated for that context and application?
Okay, so we're talking about you have one that shows no knockout, but the biological model works. The knockout, in a way, will probably trump the biological validation unless you're looking at a different cell line. So I'm assuming we're talking about same application, same cell line here. So if that's the case, then yes, you have a cross-reactivity somewhere that you've now uncovered with the knockout. And again, it could be, we've seen that for like AKTs, because the homology can be very high. Again, it might not be that you need to completely take it off market, but you need to really qualify, is it cross-reacting with the other AKTs, for example. So yes, we have come across that, and we've changed nomenclature then in the catalog accordingly.